I'm going to talk today about the neurologic complications of COVID-19. And let's see, there we go. So I thought I'd just do a brief introduction. The coronaviruses, we've all heard a lot about them in the prior months, but we know that they infect humans and numerous animal species. And while we're well aware about their effects on the respiratory tract and the GI tract, less has been discussed about neurological disease. So as you know, there are multiple coronaviruses that can infect humans. List 7 of them here, but 3 of these have previously been shown to infect neurons, including SARS-CoV-1. And I'll show you a little data that were generated about the human coronavirus OC43 a little bit later in the talk. And no COVID talk would be complete without talking a little bit about the influenza pandemic, the H1N1 pandemic from 1918. And so all of you are very familiar with that as well, but we don't typically talk very much about the neurological syndromes that occurred during that pandemic. We know that a lot of people were killed, sort of in comparison to what we're experiencing today. It wasn't people who were older who were at greatest risk for mortality. It was people between 20 and 40. And there were a few theories about this that were built upon when archived virus in people who were found in permafrost was sequenced and they could study it more. But part of the increased mortality actually may have been due to something called Encephalitis Lethargica. And there were a few forms of this, and symptoms commonly included somnolence, ocular gyric crisis, and behavior disorders. And it was first described, it's attributed to Konstantin Baron von Economo in Austria. And the paper there on the right shows the initial report. And you'll notice it's actually from 1917, so a little bit before the influenza pandemic began. And so it's been considered whether or not this is truly attributable to H1N1 or not. But it's an interesting controversy. It's unresolved, but at least we can say that there were many cases of encephalitis lethargica, estimated more than 1 million, and these 3 predominant types. And about 1/3 of people died acutely, about 1/3 recovered, and the remaining 1/3 developed sequelae, most often post-encephalitic Parkinsonism. So if we focus on the 3 coronaviruses that have had outbreaks in the last 20 years, SARS-CoV-1, MERS-CoV, and SARS-CoV-2, you can see they've all had reports of neurologic syndromes. And as I mentioned, there are data supporting from SARS-CoV-1 and MERS that neurons can be infected, and that and with SARS-CoV-1, there were actually SARS sequences isolated from brain tissue collected at autopsy. Whereas this particular summary characterized the neurologic complications from SARS-CoV-1 and MERS-CoV as sporadic, up to 34% of hospitalized patients have been reported to have some neurologic complication, and there have been sporadic, of course, case reports as well. But since we're still, doesn't seem like we're early in the pandemic, but we're still relatively early. There has not been a lot of research yet looking at on the neurologic complications and looking at virus from brain tissue and CSF. Early on from Wuhan, though, there was this highly cited report and talking that summarized symptoms and findings from people who were hospitalized with SARS-CoV-2 infection. And this particular summary table separates them into people who were considered to have severe manifestations and non-severe manifestations. Again, all of them hospitalized. And I've just excerpted out here the neurologic symptoms, effects. And what you can see is that among hospitalized patients, which of course are just a minority of people who have SARS-CoV-2 infection, that a substantial minority have CNS effects. So about a quarter of people had CNS effects, about 31% severe and about 21% not severe. And you can see it's a rather broad spectrum of neurologic effects, dizziness, headache, impaired consciousness, and more concerningly, even things like stroke and ataxia and seizures, although the last two were not very common. What is well publicized are the effects on taste and smell. And you can see here, though, that only affected about 5% of hospitalized patients. And I didn't find very many analyses looking at risk factor for neurologic effects, but this study looked at what correlates were, and people who had CNS symptoms, they had fewer platelets, fewer lymphocytes, higher blood urea nitrogen, and possibly higher creatinine. So all of those are consistent with people who have more severe disease, and that may be simply identifying that people who have more severe disease are more likely to get nervous system complications. So, if we just make a list of these complications, or borrow this pretty colorful figure from one of the reviews I looked at, the most common seem to be, in general, hyposmia, hypogeusia, and headache. And less common are delirium or impaired consciousness, so it was a bit more common in Wuhan, but overall a bit less common in the entire group of people with COVID-19, not just hospitalized patients. Stroke, but both ischemic and hemorrhagic stroke. Seizure, polyneuropathy, including acute inflammatory demyelinating polyneuropathy or Guillain-Barré. Cranial neuropathy, acute disseminated encephalomyelitis, and I'm also interested in this idea that brain involvement could actually contribute to reduced respiratory drive that could help lead to, along with severity of pulmonary disease, the need for intubation and mechanical ventilation and difficulty with removing people from ventilators. So involvement of the brainstem and respiratory impairment, as you can see in the colorful diagram there as well. So hyposmia and hypogeusia, again, the reports vary widely. I showed you in the table the prevalence in Wuhan. In Italy, it was nearly 20% of people had one or the other. And in one report, or case series in Germany, more than 85% of people had olfactory and gustatory dysfunction. So it's very, very broad estimates of prevalence in these different case series. And it probably has to do with with the characteristics of the patients in the case series, but also how the data were collected. So, people are advocating, including us, that there be some sort of standardized testing or dedicated testing to looking for these conditions. And David Moore at the HNRP, many of you know David, was already evaluating smell tests as an indicator of cognitive impairment. It's used in Alzheimer's disease. But looking at that in people with HIV, but was already developing these methods when COVID came around. So one of the very concerning syndromes and sort of a catch-all category is impaired consciousness, and many things can contribute or be manifest as impaired consciousness. The most common we think about is delirium or toxic metabolic encephalopathy. Everyone knows the risk factors for delirium in hospitalized patients. They look very much like the list of risk factors for severe COVID-19, including advanced age, multiple comorbid diseases, poor functional baseline, severe medical illness, malnutrition, as well as existing cognitive impairment or mental illness. It's thought to be due to sepsis and cytokine storm, that being an important factor contributing to the development of delirium, among others. And several inflammatory mediators have been implicated, some of which have also been implicated in severe SARS-CoV-2 infection. So IL-8, IL-6, IL-10, TNF-alpha, CXCL10, and others. Seizures, I thought would have been more common, but there's one case series that found that only 2 people out of about 300 had seizures. But what may be more concerning is that there's some evidence that COVID-19 infection can lower the seizure threshold in people who already have a seizure disorder. And so while that's not a large, very large group of people, it is a vulnerable group of people who I think we don't usually think about as trying to particularly protect from getting COVID-19. And then, those of us who are particularly interested in central nervous system infections, are very interested in whether actual encephalitis due to SARS-CoV-2 occurs. And there were case reports of encephalitis with SARS-CoV-1 and MERS-CoV, but there aren't very many data at this point that support that true encephalitis occurs with SARS-CoV-2. It certainly doesn't seem to occur very commonly. I think very concerningly were these reports of stroke. These are, terrible to occur to our patients at any sort of demographic, but this table summarizes 5 patients that were just shown briefly in the New England Journal of Medicine that had strokes from New York who were all younger than 50. In Wuhan, about 5% of hospitalized patients had acute stroke, and older age was a risk factor. And these 5 patients, apologies to Alan McCutcheon if he's on, I know he does not like me to put small text tables in slides, but hopefully they're not so small that you can't read them. But you can see at the top that, again, by definition in this series, all of them were less than 50. But you can see one patient all the way at the right is 49 years old, and the youngest, these are ordered by age, the youngest at the left is 33. And all of these were ischemic strokes, and but their syndromes varied, again, quite broadly. Not the same distribution was involved. You can see at the bottom some of their lab indicators. There was not a lot of consistency in those. Their COVID symptoms were not very consistent, and so they all had less than ideal outcomes, but there was not a lot of consistency to identify who might be at particular risk for these. The thought is that these people, though, had more severe disease, and so they had so-called cytokine storm, so very robust immune activation and inflammatory response with endothelial activation, which seems to be, I'll come back to this point, seems to be a very important event for a number of end organ complications in COVID-19, as well as hypercoagulability, which can occur with substantial immune activation. So I also mentioned AIDP, or acute inflammatory demyelinating polyneuropathy. There's another case series of 5 people with Guillain-Barré syndrome, another sort of terrifying syndrome, I think. Sorry, Siri is speaking up in the background when I said terrifying syndrome for some reason. It typically occurs about 5 to 10 days after onset of COVID-19 symptoms, and you can see that here. The patients here are oriented in rows rather than in columns as in the prior table, and you can see that there's, again, not a lot of consistency in CSF findings. They did not have anti-ganglioside antibodies, or not a lot of consistency or findings on MRI. So they tended to have high proteins in CSF, which is typical of Guillain-Barré. And I'll come back to this point in a few minutes that, when we think about diagnostic tests, mostly they track with what the clinical syndrome is, such as here with Guillain-Barré and a high protein, more than with something particular to COVID-19. So there's also a couple of other case reports, including a Miller-Fisher variant of Guillain-Barré in the literature. So we don't want to forget neuropsychiatric complications either, and this is a nice summary article also I'd refer you to. These, you can see the list of conditions here, again, include impaired concentration, but also anxiety, insomnia, memory problems, depressed mood, and so on. I won't read down the entire list, but you can see that the point estimates here, the prevalence estimates are not small. 42% for sleep disturbances, 36% for anxiety, impaired concentration or attention, 38%, and so on. So these are also important conditions. And this particular analysis raised this point that I think is also very important about the persistence of these conditions after the acute illness has resolved. So in people who survive, the prevalence is much lower, 42% versus 12%, but the fact that they persist in 10 to 20% of people, even after they recover, is troubling. And the timing at which they're assessed is important. So we haven't had very long to assess people after their recovery, only a few months. But so we'll see whether or not the persistence resolves after a longer period of follow-up. But it at least raises this point that we have to be mindful about neuropsychiatric complications from COVID-19 after people's acute symptoms have subsided. So Tom Solomon and others also published a very nice summary, and they attempted to look at the timing of onset of these different conditions. And while this is a nice effort, I don't find this graph particularly informative. If I place myself in the hospital and I'm trying to figure out what someone has based on the course of their illness, you can just sort of see this monolithic group of conditions here: Guillain-Barré, cerebral vascular disease, para-infectious, post-infectious vascular, all occurring in this 2-week period. So, what's maybe a little notable is that there are reports of Guillain-Barré and apparently some cerebral vascular disease reports when people were only RNA positive, but before symptoms came on. So I think that's an important point. Also, they've got encephalitis here too. They don't grade the strength of the evidence, so I don't really know how strong the evidence is, but they also indicate reports here of myelitis and acute disseminated encephalomyelitis, things that we typically think about as post-infectious. And so there are post-infectious complications that occur as well after resolution of the acute syndrome. ADEM, acute disseminated encephalomyelitis, is sort of a classic central nervous system post-infectious syndrome. But with SARS-CoV-1, there were also reports of post-infectious Parkinsonism as well. So that's another concern, but there haven't been many reports of that yet. Michael Benedict, who trained with Tom in Liverpool, also published this nice series, and they looked at cerebral vascular versus neuropsychiatric complications. And I thought this was a nice graphic that showed that, not surprisingly, that in general, the cerebrovascular complications occurred in people at older ages, although again, there are these very troubling strokes in young people, but the neuropsychiatric complications tended to occur more in people who are younger. So that's another interesting finding that could inform our understanding of how these different complications are being caused. So what about diagnostic assessment? You know, again, it really, in my opinion, it ought to be driven by the clinical syndrome. And so whether someone has delirium or possible encephalitis or polyneuropathy, you ought to be evaluating them based on that. And in fact, Tom Solomon's paper had a very, 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 very long table that if I tried to put it in a slide would have made Alan's head explode. So I just basically tried to excerpt what I was seeing, the trends I was seeing in the table of case reports. And, you know, there was I didn't see any real pattern here. Many people had elevated protein. I don't think anyone would be surprised if someone came into the hospital with altered sensorium and we did a lumbar puncture and they had COVID and we did a lumbar puncture and they had an elevated protein. But what do we do with that? So they had normal leukocyte counts generally, so it may be informative in its absence of pleocytosis. And some had elevated opening pressure, but there were very few reports of people measuring SARS-CoV-2 RNA or antibody in CSF. And so I think that's an opportunity, but right now I didn't see any reports that would help us identify whether someone's got a SARS-driven central nervous system complication based on their CSF findings. Similarly, there's several reports of MRI findings, but they seem very much linked to what the clinical syndrome is. So here on the right, you can see someone who had bitemporal encephalitis, particularly the mesial temporal lobe here, bilateral and symmetric. And so things like that can occur. This person didn't seem to have HSV encephalitis. But also this report of CNS inflammatory vasculopathy here at day 1 and here an evolving lesion at day 2. And so, you know, very severe disease. People will very likely have long-term sequelae from this severe disease, but it doesn't seem to happen in a high proportion of people with COVID. And then EEG also, some people reported evidence of epileptogenic waveforms on EEG, and certainly that would be important if present to put someone on an anti-epileptic drug, but I would think that we probably would have done that had we seen MRI findings like this anyway. All right, so there was this single center MRI case series looking at a single center, 2,200 patients, 112 of whom were referred for neuroimaging. You can see the indications for imaging in this bar graph here: mild non-focal symptoms, stroke, TIA, traumatic brain injury, and so on. And the green is normal imaging. You can see not all of them had MRI. Most had CT. As you could imagine, someone coming in with traumatic brain injury into the hospital probably got a screening CT in the ED. But even people who had non-focal symptoms or focal symptoms, the majority had normal imaging. The exception, not surprisingly, being people whose indication was stroke or TIA, and those people were much more likely to have imaging findings indicating either a large vessel, a small vessel stroke, or a hematoma. So again, not very surprising data, but data. So if of course I have it, if SARS-CoV-2 does directly infect cells in the brain, is the entry receptor for SARS-CoV-2 expressed there? And in fact, it is. Many of you have seen those graphics of the distribution of ACE2 in the body, and it almost, when I've seen it, it shows that there is ACE2 in the brain. This is a little bit of a nicer indication showing the regions of the brain in red here that are most densely, seem to most densely have expression of ACE2, and therefore may be more vulnerable to infection by SARS-CoV-2 if the virus reached those areas of the brain. And Serena Sputich, who I admire greatly in our field, she's also wrote a very nice summary with some beautiful graphics like this one here. And it recapitulates what's in this publication here, showing that the motor cortex has a particularly high expression of ACE2 receptor, as well as several other areas, including the ventricles, the substantia nigra, and and of course the olfactory bulb. The neurons, as well as the glia, all express ACE2. So, in contrast with HIV, that doesn't infect neurons. It seems like neurons, at least based on their expression of ACE2, would be susceptible to infection by SARS-CoV-2. So this also, you can tell the graphics are also from Serena's group, very nice graphics, but this reads a lot like, just taking what we know about encephalitis and HIV, neuro-HIV in general, and applying it to SARS-CoV-2. So you can get infection of brain cells via endothelial cells, or carried in immune cells via Trojan horse-like mechanism. So these are very typical mechanisms that we think about with HIV, and they seem very apropos to SARS-CoV-2, and there are even stronger data than with HIV on SARS-CoV-2 directly infecting endothelial cells. And reviewing this literature, I tend to think that this is a very important event in SARS-CoV-2, that this much greater involvement of not only endothelial cells throughout the body, but the brain microvascular endothelial cells, with resulting activation and expression of adhesion molecules and alterations in coagulation. So I think that that's important, and would also give access of the virus to astrocytes and neurons that are around blood vessels. So this is a little different from HIV in that since we don't think that neurons are infected by HIV, this mechanism could be more specific to SARS-CoV-2. And it's been suggested that this would be a way for SARS-CoV-2 to gain access to the brain, would be to infect the olfactory neurons and then be spread to other neurons through this transsynaptic transfer. There's some data that have tried to establish whether or not that actually occurs, and they've not been supportive that that does occur with SARS-CoV-2 in people. So right now it's a theory. It's a theory based on knowledge of other viruses, but so far we don't have good confirmation that this is actually occurring. The olfactory neurons can be infected, but spread from those neurons to the rest of the brain has not been established. But as Alan would say, is it the evidence of absence or the absence of evidence? And there's a little bit of evidence of absence, but mostly it's the absence of evidence. So Susan Morgello, also a very important investigator in neuro-HIV, also published a nice summary on SARS-CoV-2. And, I just put the table in here again and the reference so you could go back and look at it. But what she does is use her vast knowledge and experience and supporting literature to postulate what different mechanisms are for different manifestations of different complications of SARS-CoV-2 infection. And so she shows very nicely here, or summarizes nicely, that a point that I want to make, which is that these different clinical syndromes likely have somewhat different underlying pathogenesis. And again, it makes it difficult to lump all of these into one category and say the virus is causing these by this limited series of signaling pathways and biological steps, and they're all very different. And so we have this wide range of complications in the nervous system occurring in people with this infection, but some of them are more directly related to the infection, and some of them are secondary to the infection. So again, I won't spend time reading through this table. I just wanted to make that point, which I think is self-evident to some degree, but I think warrants reinforcement. So I mentioned the human coronavirus OC43, that I'd come back to that, and this is, so I am. And so this is one series of experiments that were reported with this human coronavirus using several different cell lines that reflect different glial cells, astrocytes, oligodendrocytes, microglia, and neuroblastoma for neurons. And the U87 cells are astrocytoma cells, and you can see here changes, vacuolization, and inclusions in these cells, as well as in these neuroblastoma cells, and cells that were exposed to this human coronavirus, showing that they were infected. And then there's also the real CPE, cytopathic effect, not that other CPE, showing that that there's a cytopathic effect in the cell cultures when you infect them. And here, looking at virus yields, and interestingly, we know that, again, in HIV, that it's probably brain macrophages and microglia and maybe astrocytes that are contributing to nervous system complications over time. Here, these astrocyte cell lines were able to replicate virus, but you can see neurons were able to, but with successive cell passages, they seem to lose the ability to. So even in these cells, neurons don't seem vulnerable in the same way to this human coronavirus as glial cells are. This paper also looked at acute versus persistent infection, and you can see that some of the cells supported persistent infection, which, again, this is not SARS-CoV-2, but it raises concerns about persistence of coronaviruses in the brain. So still sticking with this virus for one more slide, we can also look at animal models. And this study looked at black 6 mice, black 6 mice versus BALB C mice, and showed that they could be infected with this human coronavirus, but they also differed. This is looking at survival. The black 6 mice were much more vulnerable to mortality with this virus than were the BALB C mice. And they also differed in the amount of virus that was in the brain. And you can see that actually days post-infection, the BALB C mice controlled the virus better, whereas the black 6 mice did not. And there were, again, infection of neurons here and microgliosis. And interestingly, they gave them cyclosporin A, well known to many of us, but an anti-immune response drug. And the higher the dose the animals got, the more likely, here the black 6 mice, the more likely they were to survive. So again, this is not SARS-CoV-2, but this series of experiments supports that coronaviruses can affect the brain that the effects differ in different types of animals, and that anti-immune response, that immunomodulators improve survival. So some potential lessons for SARS-CoV-2. So for SARS-CoV-2, there are a couple of ACE2 transgenic mouse models. So in these mice, they don't express ACE2. So here, with CRISPR-Cas9, they knocked in human ACE2, and this is a different transgenic. This one from Wuhan, this one from Beijing, I think. In this transgenic mouse, they found very high levels of viral RNA in lung tissue, and you can see much lower levels in brain. But there were 4 animals that did have high levels, you can see as high as in lung, in brain tissue. So again, some animals were not affected, some were. And this is showing SARS-CoV-2 antigen, not only in lung, but in brain tissue as well, supporting that the virus was replicating there. In this CRISPR-Cas9 knock-in ACE2 mouse, they showed that there could be SARS-CoV-2 RNA in the brain of these mice, and again, it seemed to be lower than in lung, which is I'd say expected, not surprising. But there also seem to be some differences between younger mice and aging mice. And I'm going to come back to that point in just a moment. I just want to point out, I'm sorry for not showing you all of the panels here that led to this merge, but it's already a busy slide, so I didn't want to make it more busy. But this is showing where the S protein was in brain tissue. This is merging immunostaining sections for neurons, astrocytes, and microglia all into one image. And the investigators, the scientists, concluded that there was SARS-CoV-2 in all of the cells in this image, but I'm not good enough to confirm that independently. It's peer-reviewed, but I can't see that for sure, even when I looked at all of the images. So this idea of younger versus older vulnerability to central nervous system effects is something that's come up a few times, and seems to there's some interesting data for it. So I'll come back to that point. So most work, one point I wanted to make in the prior slide was that one issue with animal models, right, is that there's no animal model perfectly reflects the entire spectrum of a disease in humans. And so just a reminder that we consider animal models only for what they can potentially teach us about certain aspects of the disease. And so these transgenic mouse models show us that if there is ACE expressed in brain tissue, virus may be there, but that may not generalize to people. The other point I wanted to make was about this older and younger. It's challenging to map animal ages onto humans and to say what constitutes an older animal versus what truly constitutes, what's a younger animal versus what truly constitutes an older animal. But still, despite those limitations, they still saw differences between older and younger animals. And that was true with this Syrian golden hamster. So this is an animal model that Chip and others have talked about, and seems to be a fairly good animal model for SARS-CoV-2. It's not a transgenic animal. These are just some data showing that you can infect these hamsters with SARS-CoV-2, and whether you infect other animals by placing them in the same cage or by exposing them to fomites, other animals can be infected and show a similar pattern in SARS-CoV-2 viral levels. You see N protein in pneumocytes, lymphocytes in lungs, and interestingly, N protein in olfactory neurons as well. But as opposed to the transgenic animals, where they saw a virus in the brain, there's no SARS-CoV-2 virus or pathology in the brain of these animals, although they were looked at here only 5 days post-infection. This shows, I think Chip has shown this graph or a version of this graph before. It shows the amount of SARS-CoV-2 RNA in different tissues from these animals at different days post-infection. And you can see the brain is low, much lower than lung, lower than many other organs, but measurable. And interestingly, as in some other organs, it seems to actually come up at 4 days post-infection, but then come back down. So you can see here in brain at 7 days post-infection, at least in this animal, it's the lowest value of any others. And in some of these analyses as well, there were also differences in younger versus older animals. However, that could be reliably defined. But here, this is showing data from animals that were either exposed to high doses of SARS-CoV-2 or low doses. These are both younger animals, and I can see, I'm sorry that I left out the x-axis here, but this is the brain here. Again, lower than the other tissues, but there was no virus in the brain of older animals. So again, sort of some age effect seemingly in this animal model as well. All right, that's all the animal models I'm going to show you. There's not a lot of data on human brain tissue from autopsy. The National NeuroAIDS Tissue Consortium is working to try to remedy that. David Moore and Chris Akeen lead that effort here in San Diego. I mentioned Susan Morgello. They obviously had a lot of cases in New York, and Susan, even at the advanced stage of her career, is still working harder than ever and is trying to learn more about this process. This shows one person who died, and the authors oriented these to try to tell a story. And here we see vacuoles in brain microvascular endothelial cells supporting that they were infected. And then this shows that there were blooding of viral particles from these endothelial cells, and then vacuoles with viral particles in endothelial cells adjacent to a neural cell, and then finally vesicles with viral particles in neural cells. And they repeatedly referred to neural cells. I wanted to believe they were talking about neurons, but they kept saying neural, and I couldn't see that they were stained with a particular stain to identify them as astrocytes or macrophages or neurons. So I'll stick with neural cells. I don't know if these were neurons or not, but they're adjacent to brain microvascular endothelial cells, so presumably, probably astrocytes. Servio Ramirez from Temple has done some very nice work. He does very nice work in HIV and in other fields with the blood-brain barrier. And again, I'll come back to the comment I mentioned earlier, which is that I think that the endothelial cells are very important in SARS-CoV-2 disease in general, and maybe particularly in the central nervous system. And Servio does this very nice work doing these three-dimensional reconstructions of blood-brain barriers, and then being able to expose them to various conditions, experimental conditions, to understand how those conditions affect the blood-brain barrier in a much more detailed way than other models can. And so he did some work which is not yet peer-reviewed, so warning. But he showed that the spike protein, particularly the S1 subunit, promoted loss of barrier integrity in this model, and that they triggered a pro-inflammatory response in these cells that contributed to altered blood-brain function. So we know having increased blood-brain barrier permeability, particularly substantially increased, can adversely affect the brain all by itself, but it would also ease viral access to the brain and access of inflammatory mediators. I just thought I'd show one nice little graphic and a reference for your future reference for this idea of the brain-lung crosstalk. I was debating whether to leave this in because I think our critical care colleagues would say, of course, the brain influences, respiratory failure and whether or not you can wean someone off the ventilator. If someone's had a stroke or they've got some other, they're having a seizure, they're going to need ventilation, and it's going to affect their ability to get off it. But the reason I left it in is that it brings in SARS-CoV-2 and this idea that the cytokine storm leads to CNS hyperactivation, which leads to downregulation of ACE2 in the brain, which, and that can lead to pulmonary edema, neurogenic pulmonary edema. But this idea of both adversely affecting the respiratory drive while potentially protecting the brain from SARS-CoV-2 because of ACE2 downregulation, I thought was thought-provoking. Don't know if it's valid. It's just an idea, but I thought it was very interesting to think about, and so I left the slide in. So what about the CNS safety of SARS-CoV-2 treatments? Well, remdesivir is a prodrug, as you know, for an analog of adenosine, and there's few data on neurologic adverse events for remdesivir, but its molecular weight, because it's a prodrug, is actually sort of large. It's in the range of HIV protease inhibitors, which don't get into the brain very well. Its log P or lipophilicity, its oxalate water partition coefficient, is actually pretty low, which also supports that it's not going to reach the brain in very high concentrations. And to boot, it's also a PGP substrate. I looked at whether or not the prodrug, the pro-moiety, is cleaved outside the cell or inside the cell, because if it's like TDF, it's cleaved outside the cell, right? And so it circulates as tenofovir. So here, if it's cleaved outside the cell, it's circulating as this large prodrug. If it's cleaved inside the cell, it's circulating as the prodrug. If it's cleaved outside the cell, then it's only circulating as adenosine and will get into the central nervous system. So what I found was that it's actually cleaved inside the cell. Sorry for my confused wording about that. It's cleaved inside the cell, so it's circulating as this prodrug, and therefore is unlikely to get into the central nervous system in this form, at least with an intact blood-brain barrier and blood-CSF barrier. Dexamethasone does get into the central nervous system well. It has obviously potent anti-inflammatory effects, but it also has many neuropsychiatric adverse events, including depression, sleep disturbance, irritability, seizure, stroke, and others. And then I don't think any of us is going to be using hydroxychloroquine very much, but it does have neurologic adverse events that we don't talk about very much, including psychosis, irritability, neuropathy, and neuromyopathy. Also, it can lower the seizure threshold, interacts with several anti-epileptic drugs, including lacosamide and lamotrigine. And then I've seen the recent data on tocilizumab, which were not supportive. So again, I don't think that anyone's going to be using that, at least in the short term. But there are also neurologic adverse events, and it also poorly penetrates into the central nervous system. So I thought at least I'd comment on those. I wanted to end with comments on long-term complications. Again, I made this point, but just to reinforce it, patients with stroke, encephalitis, severe complications will likely have residual sequelae after they recover from disease. Patients with milder disease may not, but could still suffer from PTSD and other mental health disorders. And this review of neuropsychiatric complications of COVID from Rogers et al. Also included this meta-analysis of point prevalences of anxiety, depression, and PTSD here. And you can see in PTSD, you can see these studies too were pretty consistent with each other. The other two were consistent with each other, but pretty broad estimates here. But between 20 and 50%, with an overall point prevalence of about a third of people have PTSD following hospitalization with SARS-CoV-2. So that's pretty high. And but you can see still 15% with depression and 15% with anxiety. So the larger burden of disease may actually be in people who are just COVID-19 affected rather than infected when we look long term, because they're going to be long-term consequences in terms of social isolation, stress, and addiction over the coming months and years. So in conclusion, the spectrum of neuropsychiatric complications occurs in COVID-19. The most severe are life-threatening but uncommon. Less severe are more common, and their persistence is uncertain. Longer-term follow-up is warranted. The pathogenesis of many complications are likely due to robust immune and endothelial responses, in my opinion, more so than that the idea that virus is being transmitted transsynaptically from the olfactory nerves into the brain, but it's absence of evidence mostly. Hyposmia may be more likely to be due to infection of olfactory neurons. If SARS-CoV-2 does replicate in the human CNS, the efficacy of therapies is uncertain. And I ended up not talking about vaccine development and implications for the brain in terms of things like ADEM, acute disseminated encephalomyelitis, that can follow vaccination. But currently, as far as I know, there's no CNS safety signal that's been reported for the vaccines that are in development. So just like to thank everyone who's contributed over the years, including, of course, the AVRC and the HNRP, support your local research center, and I'd be happy to take questions.
Hi, Scott. Can you hear me?
Yes.
Okay, good. Sorry, I had trouble in the beginning. Zoom wanted me to restart my computer, and there was no time. So I was just going to say, if I had the opportunity, that Scott needed no introduction, and that I was excited to listen to his talk because of his experience and expertise in neuroaids, and also that he's a great speaker. Saying this at the end of the talk, I don't need you to believe in what I'm saying, but you've already heard and heard his great talk. So thank you. So I'll start off with, there are a few questions. People are commenting that's a great talk. So about loss of smell and taste. You mentioned that these reports are from patients who are hospitalized, but these are also being reported in the outpatient setting. It seems to be, not universally present, but it is a common symptom. In addition to loss of taste, some patients are also reporting hypogeusia. So they taste the food to be more salty than, and think there's too much salt when others have not noted it. So that also seems to be an issue. The question that you, or the point that you raised was that this is probably due to the local sensory damage or inflammation rather than more of the central issue that might be present in Alzheimer's disease. So that may be something that's going on. It's also noted in patients who have more of an upper respiratory tract complaints rather than more systemic complaints. So that was one point. The second, some of the questions, or as kind of group these two questions, people are interested in what is the evidence of the virus in the CNS? Can you use the clinical assays available on CSF to detect SARS-CoV-2? And also, has it been isolated in the CSF and has it been sequenced in the CSF? Getting to the point that, is there evidence of the virus in the CNS?
Yeah, so I haven't seen any data supporting that there are high viral levels in the brain of people with SARS-CoV-2, but of course we'd only be looking at people who died. And so you have to have them consent to autopsy, which we don't do very much in academic medicine anymore, but particularly with how overwhelmed our medical resources have been. So it reinforces the importance and value of having studies like NNTC, already that infrastructure already in place so that you could more rapidly deploy autopsies in support of those ideas. So there aren't many or any, I'd hazard to say, but others can speak up and correct me if I'm wrong, but I didn't come across any reports of viral RNA, no less, sequences in brain tissue. I see Ankita asked about sequences in CSF, and there were some reports of, quantifying RNA, but I didn't see any mention of sequencing RNA in order to, like we would with HIV, to try to identify whether the virus in the central nervous system is different genetically than the virus in the lung or in blood. So again, I think part of that is that, everyone's been so knocked on their heels by this and focused so much on taking care of patients and on getting therapeutic trials run and vaccine trials that, that sort of work has yet to be done. I'll make one quick point. One slide I didn't include is that I saw our friends and colleagues from Gothenburg in Sweden, Magnus Gislin and Kai Blenau and others, Henrik Zetterberg, that they measured, they do what both they're well equipped to do. They measured neurofilament light and glial fibrillary acidic protein in blood from people with neurologic symptoms from COVID, and showed that people had measurable levels of these neuronal and astrocyte biomarkers in blood, and that they to some extent correlated with the severity of the disease. So supporting that, that astrocytes and neurons were being affected and injured, but it doesn't speak to the question of whether or not they're actually infected by SARS-CoV-2.
Yeah. I think the other question that Alan is also asking is, the effect of SARS-CoV-2 on the brain probably depends on the timing of the infection. So the acute phase, where there is the cytokine storm, and that has an indirect effect on cognition. But what about, the long-term effects, the once somebody has recovered from this acute infection? Of course, unlike HIV, we don't think there is a chronic infection with SARS-CoV-2, though we don't have definite proof that if the virus survives in the brain and if it survives longer in the brain. After the acute infection and effects on cognition, do we expect any long-term effect on cognition?
At this point, we just don't know. I would say, at least from the stress, the psychological stress, and things like PTSD, those in depression, those things, and addiction, those things affect cognition. So there will be people who are affected in that way. Whether or not there are, there's some sort of persistence of viral RNA or viral products in the central nervous system, I think it's very unlikely, but we've been proven wrong without any data. So I think data are better than no data, and there are prior studies with other coronaviruses trying to implicate them in the development of diseases like Parkinson's. That there's a coronavirus infection and either some sort of persistence or molecular mimicry and autoimmune response that leads to chronic injury of the brain. And so theoretically, it's possible, but you can tell by my tone, I'm doing everything but waving hands here, so I'll wave hands, but we're pretty far from knowing whether that's truly going to happen with, based on data. So we have to follow people. I think it's prudent. It's prudent to sample them. It's prudent to evaluate people in order to really understand whether or not that may occur after all this is over. So
yeah, so one of the things we talked about, briefly discussed earlier, was that even if we don't think this is a chronic infection, people are at risk of getting reinfected, and also once a vaccine becomes available, how does that play into this host immune response mediated effect on brain function or on cognition? Any thoughts on that?
I'm sorry, Jay, say it again. I was reading Alan's question instead of listening to you.
Yeah, so I was saying that we think that the effect of the virus on the brain is probably, whether it's direct effect on the brain or it's immune mediated, can be affected by reinfection, number one, and also by, once the vaccine becomes deployed and it is effective and deployed, also would enhance the immune response. How would that, how do you anticipate that having an effect on neurocognitive impairment or on brain function?
Yeah, so, ultimately, I think my opinion, based on what I've seen so far, is that most of the complications are going to be more due to immune effects and endothelial effects, more than the virus itself in the brain, but don't know. But I didn't include a couple of slides on antibody-dependent enhancement. Many prior speakers have talked about that. But if there is some antibody-dependent enhancement that would occur following vaccination, that could affect the nervous system. And to the extent that we worry about an antibody-dependent enhanced immune response to a future coronavirus infection after someone gets a vaccine, I think is the extent to which we would worry about that, adversely, either modestly or severely, potentially affecting the nervous system as well. So if antibody-dependent enhancement occurs following vaccination, then that could have untoward consequences for cognition and, autoimmune encephalitis and so on. But at this point, it's all just supposition, I think.
Right. All right. Looks like we are 3 minutes over. Thank you again, Scott. That was a great talk. Very informative. Thank you.
All right. Thanks, everyone.